So hi everyone. Welcome to this week's single cell and spatial exploration club. So for today's session, we actually have a really exciting sort of case study to sort of share with all the scientists who use bioterrain. So it'll focus on sort of the title would be how we can go from clones and transform that into a specific context, linking T cell fate with spatial microenvironment within specifically in the context of head and neck squamous cell carcinoma. So that's going to be sort of the focus of today. Um, to kind of recap or give you some idea of what the agenda will look like in terms of what we will cover today, I'll go through sort of an introduction on some of the unmet needs within head and neck cancer and specifically relating to immunotherapy. From there, we'll go into sort of an intro to Spatial X, particularly specific advanced features that we recently released. Um, and the third part will be really going into the specific aims when we're trying to analyze a data set within the Spatial X system. So we'll be sort of using a public data set that you can actually access or request and sort of use the Talk to Data system to identify and explore these spatial public data sets. So this one is actually a really recent study, really interesting data, clinical data that you can use as well. And we sort of broken down these three aims into very specific research questions that you can answer from the features in the system. The first sort of aim will be focusing more on the immune niches versus tumor-rich zones in the tissue microenvironment. And this will be sort of explained in terms of features where you can visualize specific gene targets or the transcript of your gene within the image. Specifically, in this case, focusing on T cell clusters and how these can sort of relate to immune hubs in the tumor. We'll then go into the next aim, which will be looking at how can we incorporate neighborhood and distance analysis to measure the spatial proximity of T cells to the tumor and from other immune cells. How can we quantify and locate and shape or look at sort of exhaustion and memory states within the tissue? And the last sort of aim as well will be sort of how can we look at cell-to-cell communication analysis using our sort of internal sort of algorithms to really look at how can we map these ligand receptors that are actually driving these cells to communicate and sort of the distance of these cells to actually be closer within the tissue. So that's sort of our main focus of today. And then we'll sort of recap into some of the main summary points that we gathered from the session. Okay. So to give you a bit of feedback on sort of the unmet need within head and neck squamous cell carcinoma. So when sort of checkpoint blockade as a treatment has transformed cancer care in different types of sort of oncology case studies. But specifically, if we look at head and neck squamous cell carcinoma, we can see that within clinical data and other studies, there is still a really high non-response rate and really high fatalities from these patients. Only about 10% of these patients really achieve durable benefit. And despite that, the tumors are heavily sort of infiltrated with CD8 T cells. So we can kind of think of this as like, let's say we have two tumors, they might look identical within single cell RNA-seq data or flow cytometry data, yet they really harbor very specific structural lymphoid sort of structures within the tissue that can aggregate or preserve sort of these specific clones within sort of the tumor. And while this shows really diffuse exhaustion, we really want to look at and sort of think about this as really analyzing the architecture of the tissue. So for example, T cells near the tumor and myeloid cells might upregulate specific exhaustion markers. And we can think of this like how can this affect response from some patients that are receiving treatments, while those that are near B cell rich environments or stromal niches might retain sort of this memory sort of features within the tumor microenvironment. So that's kind of the idea that I wanted to sort of just very briefly just give you an idea of what we are going to explore today, and really trying to understand the critical unmet need when we're sort of looking at head and neck squamous cell carcinoma, and how can we distinguish immune responses from dysfunctional states in situ. So actually looking at the tissue architecture. Okay. So now, as I mentioned, I'll be sort of using a public data set that can be accessed through the Talk to Data system. So very soon, everyone who's actually using the biotrain system will be able to go to Talk to Data, select the spatial transcriptomics database, and be able to just look at what studies we have currently in the database relating to different either oncology specific disease models, different tissues, and also focusing on different technologies. So if there is a case where you really want to mine the public database to see if there's any bulk spatial transcriptomics data sets that relate to your data, or even going further into single cell resolution spatial transcriptomics technologies, then you can use and sort of utilize the Talk to Data system to access that data. Okay, so a bit of just some details on the study that we'll be using. So this study, particularly looking at TCR profiling within T cell phenotypes and clonality in these sort of human tumors. This specific study looked at an integrated or used an integrated multimodal approach. They had flow cytometry data, they had single cell RNA-seq data with the TCR sequencing profiling to look at the clonality within, and also the spatial single cell transcriptomics sort of images that actually was able to look at TCR probes within the tissue architecture. So since this data set actually has this multimodal approach, that's the reason we wanted to really look at this study. It is new, so it's from 2025, so this year. They looked at specifically around 27 patients with this sort of head and neck squamous cell carcinoma. And really the focus, as I mentioned today, is really how can we utilize Spatial X to really look at the immune niches versus the tumor zones in the database. How can we run neighborhood and distance analysis to look at sort of the measurement of proximity to tumor to other sort of immune cells within the tissue, and really looking at the mechanisms that are driving that distance and also how they interact with other cells. Okay. Okay, so just before I go into the actual live demo where I'll show you actually how you're able to create those analysis from the system, I do want to recap really what Spatial X actually is used for. So it is a platform that is specifically sort of specialized for analyzing spatial imaging data across different technologies. So really looking at multiple samples across different multi-omics data sets, which can be sort of through a side-by-side comparison. You can overlay H&E stain images as well if you want to be able to sort of map that out with the segmentation of the tissue, especially if you're working with single cell resolution technologies. And we have many capabilities when it comes to other models that you can run for segmentation. So if you want to do sort of a morphology-based annotation, or you want to look at the segmentation based on the tissue architecture or morphology, you can use Spatial X to do that as well. Okay. So some of the features that we'll be sort of covering is the cell-to-cell communication analysis, which has been recently released within Spatial X using the COMET sort of method. And this specific function, really what it tries to sort of use is be able to identify ligand receptor interactions between adjacent cells. So what we really want to try to understand is what are the ligand receptors that are actually driving that interaction. Can we actually look at the average ligand expression and the receptor expression, and what cells are actually within the actual tissue? Can we see that interaction? So that's a really important feature, especially if you're trying to look at how CD8 T cells or TCR clonality really relates to how the distance those cells are to the tumor. Okay. The next feature, which is also one of the advanced features in the system, is really looking into distance query. So what this really tries to do is understand and identify what cells are located within specific distances of a cell type of interest. So in this case, since we're looking at a study that really tried to profile TCR receptor sort of analysis, we can look at different CD8 T cells, exhaustion, proliferative specific populations, and sort of the distance or the spatial proximity to other immune cells or even tumor cells in the tissue. So that's sort of the idea, is really how can we compute that distance? What is the nearest anchor from that cell? And can we sort of use different thresholds to look at different distances? So for example, 20 or 20 to 50 So really that's sort of things that you can actually apply within SpatialX to be able to understand and answer that question. The next feature we'll also cover is the region segmentation. So this really what it tries to do is even go further into defining what are the functional spatial clusters, or what we would term spatial niches within the tissue. And this sort of feature, it kind of adopts sort of a weighted gene co-expression network analysis to really understand what are the pairwise correlations of gene expression within the tissue, and does that clustering actually relate to a network? So is there specific modules of genes that might have a functional sort of role that makes them specific, like physical adjacent to other tumor cells, and what are the pathways that are actually driving that segment or that specific region? So that's also a very important feature, and it tries to answer other types of analysis, like what pathway might be having a role with these cells that are actually sort of interacting with each other within the tissue. Okay, and the last feature that I'll try to cover is neighborhood analysis. So this actually tries to also understand the co-occurrence patterns in a very different way. So it uses a specific methodology where it tries to look at consistent patterns or proportion matrices within the tissue by looking at pseudocells and how these pseudocells sort of have this hierarchical clustering. And do they cluster within specific groups, and what is sort of that dominance, or what cell types are actually dominating those cells to have this specific pattern. Okay, so now we'll go into the SpatialX demo, which I'll sort of cover that specific study that I mentioned that had many different patient samples relating to head and neck squamous cell carcinoma, and specifically a study that actually did a really important job in being able to also manually sort of curate different clinical data. So you're able to see if the patients had treatment previously, what type of treatments that they have apart from sort of cancer-specific treatments, as well as what was the response to that treatment. So all of that specific clinical metadata is available within the study. Okay, so now I'll go into the specific SpatialX system. So this is sort of the interface that you'll sort of interact with when you're analyzing different tissue images or different architectural structure of the tissue. In this case, I want to make it clear this study is a Xenium data set. So this is a single cell resolution data set, which is quite important because it actually gives you a lot of different information. When you're actually zooming in into the tissue, you're able to actually visualize the individual cells that are segmented and overlaid within the H&E stained image as well. In order to be able to actually interact, when you're accessing any study in the database, you need to make sure that all these different preferences and actual sort of keys are actually turned on, or the different channels are turned on, so that you're able to actually visualize these different samples. So you can see here, this study has many different images. I want to make sure that I'm looking at all the images within that study. I want to make sure as well that the cell segmentation is combined so that I'm able to see it across all the different samples, whether it's primary or recurrent tumors, or if they have different treatments. For example, if we go into the metadata of the study, we can see if they have a P16 status, or we can see the sampling site of the tumor as well. Or if they had specific treatments within that data set. So all that information you're able to find within the public study, and you can directly sort of select what information you want to see within that data set, and what samples are relating to what treatments or what response rates. So that's sort of how you can start interacting and just making sure that all the layers of the image is actually turned on. One thing to keep in mind is normally the point size will be really large because you want to be able to see like the actual spots in the expression of the tissue. But if you want to be able to make that a bit smaller so that you're able to see multiple cell types, you can adjust that point size as well. Okay. So I'll go into this combined data. So you can see I'm able to see these different sort of tissue structures. Um, Samples that are coming from different treatments as well as different sampling sites. So tumors that were either primary or that are reoccurrent within that patient. And this is where we can start sort of answering the first question in our sort of analysis. So one of the first questions, as I mentioned, is really how can we determine if there's immune specific niches within the tissue or tumor-rich zones within the tissue? And in this case, since this study, again, is TCR sort of specific, I wanted to look at sort of very specific T cell markers that were relevant to like memory T cells, exhausted T cells, or even proliferative specific markers. So in order to do that, normally what you can do is directly input your specific genes that you're focusing on. In this case, I want to look at these specific markers. I can look at the gene individually within the tissue so I can see if it's actually expressed, and I can actually adjust again the size of the actual point so that I'm able to see the expression a bit clearer. So you can see here it automatically gives me the ability to see, okay, within what region do we see the specific expression of a certain exhausted T cell marker. Now, if we have multiple genes and I just want to see based on the enriched sort of enrichment zone of all of these specific T cell markers where they're actually expressed, I can look at all the genes at once and I can see where do we see sort of this enrichment pattern within the tissue and how does this relate to differences in either primary or recurrent tumors. So again, you can zoom in and you're able to get sort of that information directly, just looking at specific markers and really where are they located or localized within the tissue. Okay. Now, the other factor is as well, how can we determine the type of cell types that we have within the study or within these specific samples? So this study specifically didn't have metadata relating to the actual cell type annotation. So the author didn't provide that when they provided the actual sample data. So if you do have public data where it's limited by either cell type annotations or you want to know the proportion of different cells that you have within each of the samples. We do have a cell type prediction sort of algorithm that is able to really determine from other public data. So it tries to really mine the entire talk to data system and try to find sort of overlap signature markers that will determine whether that cluster within your study is specifically like a T cell cluster. Is it a macrophage specific marker? So that's where you can sort of go into what prediction type or what granularity you want to go into. You can also customize the reference model, which makes it more specific to the data that you're working with. So for example, in this case, this is a head and neck squamous cell carcinoma. So what I can do, I can add specifically regions or specific ontologies that are specific to the analysis that I want to perform. So for example, this is relating to tumors that are normally in the oral cavity. And there's different areas that you can add as well because this tumor might extend to different regions, including regions closer to the esophagus as well. So you're able to really select what tissues you would be focusing on, as well as what condition or what is the disease model that you're actually analyzing. So that's where we can add here head and neck, and you can add those specific studies relating to your sample. So that you're able to pull in public studies from the database that are specifically focusing on the same disease model. So now that you have these filters and you select to run the prediction results, one of the sort of, I can show you here the result that you can go through. Is that it'll automatically provide you these different cell type labels within the sample. You are able to track back and look back at the results of the cell type prediction as well. So if you're interested in looking at the full log view, or you just want to see like what samples or where do we normally see these cell types, you can go and sort of visualize the different sort of samples in the system, and you're able to sort of get more of an idea of what sort of specific tumor sampling sites have specific like tumor cells, and which ones have like the CD8 exhausted specific T cells, and what is the distance within these cells within the tumor. So that's sort of another step that will give you this metadata that you can use to run other analysis from here. Okay, so this is just an example of some of the differences based on sort of a sort of reoccurrent tumor versus a primary tumor, and what sort of cells we see within these specific tissues. So for example, in a reoccurrent patient sample, what we see is that there are a lot of specific regions where we see a high proportion of these CD8 beta T cells. Whereas if we're looking at a primary tumor, we have many macrophages in very specific regions. And this is quite different to what we see like across different samples. So that's where you can start getting an idea. What is the clinical background? What is the tumor stage? Does it make a difference in terms of the proportions of the cells? And some of the things here that we can see is that when we look at the expression of these specific genes relating to the CD8 or exhausted T cell markers, we can see that these are actually localized within cells that are B cells and stromal cells within the primary tumor. Whereas if we look at the CTLA-4 sort of target, this is mainly within these exhausted T cell regions within sort of tumors that are reoccurrent from these patient samples. So again, this is sort of initial, I guess, analysis that you can perform to understand these results. Now, the next sort of function is a region segmentation, which I was mentioning that this is more related to immune functional hubs or tumor regions that have either specific enrichment of certain markers or certain pathways that are driving that enrichment within that functional hub in the tissue. So in order to be able to run this analysis, you'll be able to see that we have the region segmentation model. It'll be able to run that spatial gene correlation analysis, and you can also adjust the radius as well to be able to identify these specific regions or any other advanced parameters that might be specific to your data. And since this feature takes a bit of time, I just wanted to show some results here on how you're able to sort of analyze this. So normally what you'll have is you'll have your sort of sample, you'll have these different segments that are related to these functional hubs from the region or the spatial gene correlation analysis. And within each segment, you'll be able to actually see what are the genes that are enriched within that segment. So it's actually quite important because instead of looking at individual markers that you might already know that are specific to like exhausted T cells, you can add an entire sort of, or look at an entire gene set and actually look at the enrichment score of that gene set within the tissue. So that's where these actions will allow you to do that. So you can actually visualize the AU cell score, and you can visualize the actual pathway that is driving that enrichment. And in this case, what we can see is that within this purple region that you can see in the figure, we have very specific immune response genes that are correlated or expressed, which are potentially within sort of the tumor region. We can also see that there's a high expression of these IFN sort of responsive genes. So for example, if we look at OAS1 or this MX1 or IFT1, these are specifically IFN induced chemokines that can also attract T cell sort of activation or sort of neighboring sort of the tumor microenvironment as well. So again, this is also a very important feature I wanted to highlight that you are able to run within the system. Okay. Now, if we go into the next feature, which is more related to how can we actually calculate and find cells within a specific distance, and can we see that within a specific surrounding or inflammatory tumor cells? So if I go back into the Spatial X system, in this case, what I want to do is actually go into one of the samples here that I have, specifically relating to these reoccurrent tumors. And what I want to do is actually select the CD8 positive alpha beta T cells because I'm interested in really seeing if there's any correlation within this exhausted specific markers and how are they close in distance to these other cancer tumor cells within the actual tissue. So when you select the actual cell type population, what you want to do is actually go into these other icons here, which allows you to run this distance query analysis. There are other features that are also quite important, which allow you to identify marker genes or the top expressed genes within a specific region. But you can simply go into distance query, and this is where you can add the specific distance that you really want to focus on for this specific cell type. So when you run the distance query, it'll calculate that distance for the tissue. It'll take a few seconds to actually load, but you'll see that it's very quick on actually showing you that information. So there's two key points that I want to highlight when you're running distance query. One, which is the number of targeted neighbors. So this is sort of this image here. And what this is showing you is actually how the CD8 T cells in the warmer orange area region lie within 0 to 50 micron of other myeloid cells within the actual sort of sample. And it's showing you this close proximity correlates and how this is increased based on either exhaustion or other related counterparts within the tissue. So again, if we zoom in, you're able to directly see the actual CD8 T cells and these other neighboring cells that have this specific distance that I want to sort of calculate. The other sort of, I guess, unit that you also need to pay attention to is the number of targeted neighbors, which is quite different. So if I switch into this image here, what this is really trying to show is how these neighbor counts highlight this density of these CD8 clusters within the actual sample. So this is where I'm actually looking at this yellow-orange sort of enrichment hub. And I'm trying to see how this specific hubs are actually located with other immune cells and these other lighter regions that are not really showing this dispersed infiltration. So these are different calculations that you can run, one which is more relating to the actual distance and how you can calculate that distance based on sort of that specific micron or the minimum distance that you input, and also the number of targeted neighbors that shows you that density within that specific region in the tissue. Okay. Now, another important part here is how can you actually save these results, especially if you want to look at, okay, I was able to identify these specific sort of regions where I see this either targeted neighbor region or the actual distance of interest. In order to do that, what you want to do is select the specific visualization and you want to create a new annotation so that it actually saves that 50 micron information or that distance labeling. And you want to create a new sort of label here. So for example, the upper metadata label could be just like niches. So you want to identify different niche structures within the sample. And then the actual label is relating to either the distance sort of I guess, input that you're adding in the system, as well as what is the cell type that you're focusing on in this case. Okay, so now that we've added this, I can create a new annotation in the system and that will save. And what I want to do is actually look at the proportions of that. So if I want to see, okay, what is the proportion of this with other immune cells so I can actually visually see it in a figure, what you can do is then go into the cell type or the chart and graph view. You can go into basic charts. And in this case, what I want to do, I want to look at the niche results that I have. I want to remove any of these genes here because I'm not really interested in the actual genes. And what I want to add is also what is the actual sort of information that I'm looking at. So in the bubble heat map, what you can do is split it by other conditions, or you can also choose to look at it as a proportion plot where you're looking at how this region within the 50 micron has specific proportions of these immune cells. So I can actually clearly see that within my CD8 T cell cluster, the highest proportion or the highest neighboring sort of distant cell is these neoplastic tumor cells. From there, the other cells that actually are also highlighted is these macrophages as well. And you'll see that these other cells are actually lower in proportion. So mainly, I can see that my CD8 exhausted T cells are neighboring within these neoplastic cells in the tumor. Okay. So that's what I really wanted to highlight is that you can actually save the different distance queries that you're running in order to be able to actually visualize them and look at the proportions of these different cells. Okay, so the last, I guess, feature that I wanted to highlight is the neighborhood analysis. So this one also is quite important because it tries to really look at the co-occurrence of these cells. So is there a pattern that we can see, not just like a one-time region? And can we actually look at this in a more quantitative way? So in this way, when you're actually going into the neighborhood analysis, this is where you're able to access the feature. You can select what is the input. So in this case, I'm looking at my different cell type annotations. I can select what radius I want to look at as well. And then I can simply run the neighborhood analysis from there. So an example of that when we go into the results is that we're able to look at these different cell types within my different samples. I can look at the correlation co-occurrence score within specific populations of interest. And I can also look at neighborhood clustering analysis. And a way to see this is that the more, I guess, away you get from the actual table, the more higher structures you have, the more inner you go into these sort of proportions, the more granularity you'll have within the spatial domains. So for example, here, what I'm trying to do, I'm trying to look at the main spatial domain, so not really granular because I haven't really subsetted specific T cell populations. And I want to be able to see what are the proportions of this sort of neighborhood co-occurrence. So I can directly see as well that also within these spatial domains, when we're looking at neighborhood analysis, I can see that there are a lot of different cell type populations that are co-occurring in specific samples that might be different if it's like a reoccurrent or a primary tumor or the different treatments that are used as well. So you can directly see that it really depends on the sample in terms of what is the proportion of that neighborhood clustering as well as the actual cell types. So this is a way that you can quantitatively be able to calculate that cell type co-occurrence in the system. Okay. So I guess this one I don't have that much time to go into, but it's also a very important thing to keep in mind is that now that we were able to see these different populations co-occurring, what are the ligand binding interactions that are actually driving that those cells to actually interact? So in this case, if you're interested in actually running that feature, you can go into the cell-to-cell interaction analysis, and these are the specific figures that you'll be able to generate. So in this case, in my data, I can directly see that these specific dendritic cells are actually communicating quite highly with my CD8 T cells. I can look at these specific ligand binding pairs that are driving that specific interaction, and I can actually visualize it within the tissue image directly. So a more clear example of that, if I zoom in into that, I can see these specific cells that are actually interacting, which cells are receiving and which cells are actually sending that information through that ligand binding pair. And I can look at it through either a interaction view or a cell pair view to look at the direction of that interaction, as well as the actual ligand binding pair that are driving that information. Okay. So I guess this was sort of the summary of the main functions that we've added into SpatialX, the advanced features that you can use for answering these specific type of clonal TCR questions, specific like niche immune hub questions, and how this relates to different tumor zones in specific cancer models. So some of these summary points are quite important, which was we were able to look at specific markers to determine what were specific immune hubs and which specific zones were relating to different tumor zones and how these different markers were expressed within the tissue. We then went into more of a neighborhood composition. So we were trying to really look at what is a co-localization of these different niches and can we see that across different cell type populations. And the last two ones are more specifically related to how can we quantitatively look at the proximity of these cells and how they actually interact, whether that's through distance query. So you're actually just interested within the tissue morphology. What is the distance, like actual unit where my cells is sort of communicating with these tumor cancer cells, and really what are the ligand binding interactions that are driving that as well. So again, these are very important, I guess, questions that you can answer. And I just wanted to showcase a example on how you're able to use the SpatialX system to do that. So I think my colleague is circulating a specific survey just to be able to gather some feedback from the researchers who have joined today in order to really understand if these features are functions that you would actually use within your specific analysis, or is there any other features that you feel are not yet available but will be very important when you're trying to run this analysis from your data. Okay, so please, if you're able to fill the feedback questionnaire, that would be very useful for us to be able to understand the different research needs. Okay, so I'll double check if there's any questions in the chat, just in case there are any. Let me just go here. Okay, so I don't see any questions currently in the Q&A. So if there are any questions that you don't feel I guess, comfortable actually going through the questions within the webinar, you can always reach out directly and we can sort of have a one-to-one session where we will go through that analysis as well. Okay. So I'll just give a few minutes for everyone to fill out the questionnaire, and in case if there are any questions or anything that you would like to ask. Okay. Well, thank you everyone for joining today's session. I hope this was useful, and I hope to see you in the next session, which will be on a different platform or a different case study. So really looking forward to seeing everyone in that session. Thank you.